Alright, bro. AQA Biology A level topic one completely patterned. Yeah, we're gonna go through all the biological molecules topic here from the start to the end. Make sure that you're certain with it. Let's start then. Alright, cool. So monomers, yeah. So the definition for a monomer, yeah, nice little plain cut definition, yeah. There's smaller units from which larger units are made. And polymers are molecules made from a large number of monomers, yeah. So you got the monomers. The monomers are the small single-use units, yeah, from which the larger polymers are made from. You get me? Polymers, yeah, molecules made from large number of monomers joined together. So poly means many, yeah. So poly, yeah, like polyamorous, yeah. So poly means many. So you're putting together bare things here together to make a polymer. You get me? So some examples here. So we got glucose here, and the polymer that glucose makes. There's different types of glucose. We'll get into that in a minute. But all you need to know is the three types of polymers that are made from glucose here are starch, cellulose, and glycogen. Yeah, obviously starch is a glucose store. Cellulose is for structure, and glycogen is for also for structure. Yeah. Amino acids, yeah, they are the monomer and they form protein as the polymer, and obviously nucleotides as well. So they form DNA and RNA, yeah. So deoxyribonucleic acid and ribonucleic acid, yeah. So condensation and hydrolysis, yeah. So to make these polymers, yeah, to make these molecules that are made of bare things joined together, you can undergo one type of reaction, yeah. That's called condensation reaction. So this is a type of reaction that joins two molecules together with a formation of a chemical bond involving the elimination of a molecule of water, yeah. So when you breathe on the glass here, you see that water is formed, yeah. So that's a condensation, yeah. So this is the definition for condensation. You need to write that down. So this is going to get you all the marks here. A type of reaction that joins two molecules together. So we're joining two monomers together. We're forming a chemical bond between those two monomers, and it's also going to involve the elimination of a water molecule, yeah. And hydrolysis is the opposite of condensation. So for hydrolysis, yeah, that's when you're going to break a chemical bond between two molecules. You've got two molecules, yeah, you're going to break that chemical bond using water. So you're putting the water that got eliminated, you're putting that back in to break that chemical bond and split the polymer into what it's made of, yeah. Alright, safe. Calm. So this is a condensation reaction here. We've got monomer A and monomer B. Yeah? As you see, they have OH and H groups there. So these OH and H groups are gonna get emitted to make H2O, and there's gonna be a chemical bond as you see there between monomer A and monomer B. Yeah? So that's called a condensation reaction. Yeah? It's condensing. It's forming together to condense. You get me? Yeah. And then here's hydrolysis. So we got reactant AB. So we got A and B, a polymer. Yeah. A is joined with B, and it's reacting with H2O, and H2O is going to break that bond between them, and A is going to have a H attached to it, and B is going to have a OH attached to it in the end, because that H2O got used to forge between that bond and break that bond, and one H got bonded to that, OH got bonded to that, yeah? So make sure you understand that that's condensation and hydrolysis here. Alright, cool. So carbohydrates here. So these are just molecules that consist of only carbon, hydrogen, and oxygen, yeah? We've got three types of carbohydrates. So we start with the monosaccharides, yeah? There's three types of monosaccharides. You can get them to write them down in a minute, but you need to know these three. So make sure you write this table out. Monosaccharides, make sure you memorize this. There's glucose, fructose, and galactose, yeah? They all have the same formula. Disaccharides is when you mix together two monosaccharides. So when I join two monosaccharides, you form a disaccharide, yeah? So the disaccharides are maltose, sucrose, and lactose, yeah? And then polysaccharides is when you mix bare of these saccharides, bare disaccharides, yeah, to form, or monosaccharides to form a long polymer chain, which is things like starch, glycogen, and cellulose, yeah. So let's talk about the monosaccharides then. So monosaccharides are glucose, fructose, and galactose, yeah. So they have the same molecular formula, so they all have the same formula of C6H12O6, yeah. And glucose is a monosaccharide that consists of 6 carbon atoms, yeah, and is the main substrate for respiration and can also be converted into other biomolecules, yeah. So glucose is what we use for respiration, and that's one of these monosaccharides. But there's three monosaccharides you need to know, yeah: glucose, fructose, galactose, yeah. Memorize that. Now let's talk more about glucose. Yeah, there's two types of glucose. There's alpha and beta. Yeah, so glucose has two isomers. Yeah, so isomers are molecules with the same molecular formula. So they got C6H12O6. Yeah, but they got a different arrangement. So they look differently in terms of the structure. Yeah, but they got the same shit going on. So effectively here we've got two isomers, alpha and beta. Yeah, you need to be able to draw both of these. Yeah, just memorize alpha and beta. You just know. So for alpha, it's going to be this kind of figure, and the OH and the OH are on the same side, yeah? So OH there, OH there, yeah? So it's like a Z isomer kind of thing. So two highest priority groups on the same side, yeah? So you got OH, OH, just remember that on the bottom, yeah? Now on the carbon 1, we call that the carbon 1 because the oxygen group is there, so that will be carbon 1 there. So on the carbon 1, yeah? the H and OH flips for beta, bro. So memorize alpha and just know that on the first carbon, so the carbon there, where my mouse is here, it will flip. So now it'll be OH on opposite sides. You get that? So you need to know how to draw both of those, yeah? So make note of that. Make sure you know how to draw alpha and beta glucose, yeah? Alright, safe. Disaccharides, yeah? So you need to know what monosaccharides, so out of glucose, galactose, and fructose, you need to know what forms together to make these disaccharides, yeah? So disaccharides are made of two monosaccharides, yeah? Di means two, yeah? So, and it forms a glycosidic bond. So remember how I said about condensation reaction, you form a chemical bond? That chemical bond will depend on what chemicals we're talking about. So here we're talking about glucose here and sugars here. So therefore it's going to be a glycosidic bond, yeah? So that's the name of the chemical bond, is glycosidic bond. So disaccharides are formed from condensation of two monosaccharides forming a glycosidic bond, yeah? So alpha plus alpha glucose makes maltose and obviously water because it's condensation, yeah? So alpha plus alpha makes maltose, yeah? Listen, when you make the disaccharide, yeah, remember to take away the H2O atom, yeah? Remember that. Okay, cool. Beta glucose plus galactose makes lactose. And then alpha glucose plus fructose makes sucrose. I put beta and alpha for these two, but you don't need to know that. You just need to know that alpha and alpha makes maltose. And then here it's just glucose plus galactose is lactose, glucose plus fructose is sucrose, yeah? You don't actually need to know like this is beta and this is alpha, but it's there just in case you want to know anyway. So maltose is obviously malt or sugar beer. Lactose is the milk sugar, bro. Yeah, and then sucrose is like table sugar, like the shit that you find in the restaurants, innit? You get me? Cool. So this is how it works basically. So we've got two monosaccharide units. So we've got sugar there, sugar there. This could be any kind of sugar. And what happens is the one H from here reacts with the OH from there, and it emits water. And then that O that's left over forms that glycosidic linkage or glycosidic bond between them. Yeah, and that's obviously a condensation reaction. You're releasing water. So that's how it works. Yeah. Alright, cool. Polysaccharides, yeah. So polysaccharides, there's three types. You got starch, cellulose, and glycogen, yeah. So they are polymers made from smaller glucose subunits. I know there's bare written here, but just trust me, go through this with me, and you'll be certain in the future, innit? This is all you need for those marks, yeah. So starch is made from alpha glucose monomer. Write that down. Make sure you know that, yeah. So starch is made from alpha glucose monomer, yeah. It has 1,4 glycosidic bonds in amylose and 1,4 and 1,6 glycosidic bonds in amylopectin. Let me just check if that. Okay, I'll show you what I mean by 1,4 and 1,6 in a minute, okay? So you need to know it has 1,4 glycosidic bonds in amylose. So amylose is a form of starch, and you also got amylopectin. So you got both in starch, yeah. Okay, cool. It's in plant cells, and it's just a store of glucose. Amylose has a unbranched helix structure, whereas amylopectin has a branch structure. It's very easy to tell between unbranched. If something has 1,4 glycosidic bonds only, it's going to be unbranched, yeah? If something has a 1,6 glycosidic bonds, it's going to be branched. So just remember that for the future. Starch is effective as it is compact and hence can store a lot of glucose without using up space, yeah? So that's an adaptation of starch. An advantage of starch is that it's very compact and so it can store a lot of glucose, yeah, without using up that much space in it. The branch structure increases the surface area as well, which allows for rapid hydrolysis to produce glucose. So whenever you need glucose in the body, the starch is gonna in the plant, the starch is gonna break down quickly because it's got that amylopectin, which has the 1,6 glycosidic bonds, which means that it's got a branch structure, which means that there's a bigger surface area for rapid hydrolysis. You get me? To break that down into glucose so that glucose can be used. It's also insoluble, so it will not affect water potential. That's a free mark at the end there, yeah? Remember this, insoluble, so it will not affect water potential. These here are all like good points here to get bare marks, you get me? Alright, cool. Types of starch, so you got amylose and amylopectin, yeah? So what I mean by 1,4 is we established that that is the one carbon, the first carbon, yeah, because it starts from that O over there. So because that's the functional group. So because that's the first carbon, that means that if you follow my mouse, 1, 2, 3, 4, that's going to be the fourth one. So that's the fourth one. This is the first one. So the linkage, the bond is between the first and the fourth carbon. The first carbon on the first sugar and the fourth carbon on the second sugar. So that's what we call a 1,4 glycosidic bond. As you see, it's unbranched. In amylopectin, we've got the same 1,4 glycosidic bond there and the same one here, but we've also got a 1,6 glycosidic bond. Obviously, that where my mouse is there, that's the sixth carbon because it goes 1, 2, 3, 4, 5, and then 6 there. You see that? So because of that, 5, 6 on that CH2. That's the sixth carbon there, the CH2 one, yeah? So because of that, yeah, we've got one carbon there bonded to the sixth carbon, where that CH2 is. That's going to create a 1,6 glycosidic bond, which is obviously a chain, yeah? It's a not a straight, it's a branch, yeah? Because it was a chain there, and there's a linkage between them, so a branch. So that's what I mean by 1,6 and 1,4, yeah? Okay, cool. Cellulose, yeah? Write this down. This is made of beta glucose. So starch was an alpha glucose monomer. Cellulose is a beta glucose monomer. And it has a 1,4 glycosidic bonds only. Hence, it's very unbranched and straight chained, yeah, which means it's strong, yeah. So it's found in the cell wall of a plant and provides structural strength, yeah. It consists of long, straight chains held in parallel by many hydrogen bonds. These chains are called fibrils, yeah, and it is insoluble and hence it doesn't affect the water potential, and the number of hydrogen bonds also make the molecule very strong, yeah. So obviously this is a very strong molecule used for structural support, yeah. And obviously my man's got that situation going on in it, so obviously it's also insoluble, so it's not going to affect the water potential as well. Alright, safe. So this is the structure of cellulose. What's going on here, bro, is you got these again. So as you see, it's just 1,4 glycosidic bonds of beta glucose. As you see, the OH and the HOH are on opposite sides. That's going to create hydrogen bonds between them, because as you see, there's H there, OH there, so that's going to create a lot of hydrogen bonds vertically, and that's going to create one fibril. And so these are going to go together. Many of these microfibrils are going to go together to create a lot of straight chains like that. It's going to be very strong, and that's going to be inside the cell wall of the plant cell. Yeah. All right, safe. Glycogen, yeah. So it's a polymer of alpha glucose, yeah. So starch, and this is alpha. Uh, Beta is for cellulose, yeah. So it has 1,4 and 1,6 glycosidic. It's found in animal cells and acts as a store of glucose. So it's just basically starch here, but for animals. And it has a branched structure that increases surface area for rapid hydrolysis to produce glucose. So the adaptations, yeah, the advantages, yeah, for glycogen and starch are basically the same. They got a 1,4, 1,6 situation, and they're both branched, yeah. And also it's insoluble, so it won't affect water potential, yeah. Alright, calm. Lipids, bro. Lipids, fam. So they are biological molecules that are made of carbon, hydrogen, and oxygen, and are only soluble in organic solvents such as alcohol. Yeah. So obviously they're non-polar, so therefore they're gonna be soluble in non-polar solvents such as ethanol. Yeah. So there's two types: triglycerides and phospholipids. Yeah. Polar. So yeah. Alright. Cool. So triglycerides. Yeah. So these are lipids made of one molecule uh of glycerol and three fatty acids. Yeah. So triglycerides, they have tri in the name, yeah, so three. So they're lipids made of one molecule of glycerol and three fatty acids, yeah. So they are formed through a condensation reaction, forming three ester bonds, and hence emitting three molecules of water. So as we talked about before, glycosidic bond, yeah, is the chemical bond that's made from condensation reaction of glucose. For condensation reaction of lipids, yeah, it's going to make an ester bond, yeah. And hence, because we've got three condensation reactions going on, which I'll show you in a minute, we're going to release three molecules of water, yeah. They have a high ratio, this is important to note down, they have a high ratio of energy storing carbon hydrogen bonds to carbon atoms, so they are a good energy store. So these carbon hydrogen bonds here, they store a lot of energy. And compare that to the main chain. So let's say I've got a CCC chain. I've got bare CH bonds that are bonded to that. So therefore I've got a lot of energy in a shorter amount, basically. You get me? And they have a low mass to energy ratio, meaning they are a good storage molecule with a lot of energy being stored in a small volume, yeah? So this is what I mean. These fatty acids are going to join onto the glycerol by condensation reaction to form these three ester bonds, and it's going to release those free water, yeah? Due to the high ratio of hydrogen oxygen atoms, when triglycerides release water, they release water when oxidized, yeah? So when triglycerides are oxidized, because it's got hydrogen oxygen atoms, yeah, that HO is gonna oxidize to therefore form water, yeah. Because they are large and non-polar, they are also insoluble and hence does not affect water potential. Cool, because they only dissolve in ethanol, yeah. Safe. So here we've got it shown again, bro. So we see the fatty acids here. So that's what fatty acids there, and as you see, OH is going to join onto the H of glycerol. That's going to be one condensation reaction, creating this ester link, this COO link, and it's going to release three water molecules. Yeah, one for each fatty acid. Yeah. Okay, cool. Saturated and unsaturated. Yeah. So saturated lipids. Yeah, they don't contain any carbon-carbon double bonds. Yeah. So they're just straight chain, bro. Just bare chain. Yeah. Just pure saturation. Yeah. No like they call them kinks, yeah. No kinks in between the thing. No, nothing like that. It's just straight chain. Yeah. Alright, calm. And so therefore they have a high melting point, yeah, and they are solid at room temperature. These are things like animal fats, yeah. You know when they start, when you get to the upper echelons, yeah, they start mixing the beef tallow. They start using them things there. Obviously, the beef tallow that's gonna contain what I'm saying, that's animal fat. That's why it's just solid. You gotta mix that on the pan, to create the moisture. You get me? Whereas unsaturated, yeah, like carbon double bonds, yeah, and they cannot pack together that tightly since they got that little kink here. That's what they call it, yeah. Um, and so hence the liquid at room temperature, yeah. So these are things like vegetable oils, like the shit that you cook with, yeah. We're out here with the beef tallow shit, you get me? You man are still on that vegetable oil thing. Yeah, level up, bro, level up. Alright, cool. Phospholipids, yeah. So these are made of one glycerol and one phosphate head and two fatty acids, yeah. So we've got one glycerol there, we've got a phosphate head, which is negative, and we've got one saturated fatty acid and one unsaturated. You need to know that structure, yeah. So the two fatty acids are bonded through an ester bond formed by condensation reaction, and hence two waters will be emitted. Now, the head is hydrophilic, yeah. So that means that it loves water, yeah. So due to the negative charge on the phosphate group, so because of that negative charge on the phosphate group, it's going to be attracted to water, yeah. And the fatty acid tails are hydrophobic, so they hate water, yeah? Because obviously they're fatty acids, yeah? So hence they form micelles when in contact with water. So when in contact with water, they form like these micelle groups, like these bubble kind of things. The head does not bond well with fats, however, the tails do. So the tails will bond well with fats, but the heads do not. And the cell membrane, this activity forms a phospholipid bilayer, which we'll go on to in the future. So all that means basically, as you see in the bilayer, the heads are attracted to water. So you can imagine there's water on the other side and the outside. The heads are hydrophilic, so they're attracted to that water. And the tails are hydrophobic, so they shy away from that. So that's why it creates this kind of layer in the cell membrane, yeah? Alright, cool. Proteins, yeah? Amino acids are the monomers that make proteins. So you need to know this structure here of the amino acid. You've got the amine group there, the NH2. You've got the normal C standard. You've got the R group, which can change to 20 different R groups. That's the variable region. That's what forms different amino acids like glycine, like whatever. And you got the carboxylic acid group on the other side. So it's pretty simple to draw, not too techy. Yeah, just CNH2COOH. Calm. Amino acids are monomers that make proteins. Amino acids contain an amino group, carboxylic acid group, and a variable region. Yeah, man's mouth is moving crazy. Yeah, there are 20 different amino groups which are determined by the R group. Proteins are formed through a condensation reaction between the amino and the carboxyl group. Yeah, where a molecule of water is emitted. Yeah, so obviously, as you see, these two glycines, yeah, are forming a dipeptide. Yeah, and what's going on here is the glycines there, the OH and the H are moving together and they're all getting emitted. So the OH of one and the H of one is getting emitted to make H2O, yeah, that's a condensation reaction. And there's going to be a peptide bond now between the C and the N. So again, the chemical bond for protein formation, for dipeptide formation, is going to be called peptide bonds, yeah. So little recap, yeah, we had glycosidic bonds here for glucose. We had ester bonds for esters, for fats. And we had peptide bonds for proteins, yeah. Alright, cool. Say on, bro. So proteins got different structures, yeah. So we got the primary structure. The primary structure of a protein is the order and number of amino acids in a protein. So you need to remember that they love asking this, bro. They love asking, yeah, what's the primary structure of a protein? So the primary structure of a protein, yeah, is the order and number of amino acids in that chain. This contains the initial sequence of amino acids and will determine the protein's end function. So any change in this will change the function in the end, yeah. So calm. Secondary structure, yeah? So the secondary structure is the shape that the amino acid chain makes, resulting in either an alpha helix or a beta pleated sheet, yeah? So the secondary structure is when this primary structure starts folding in different ways. So connected with these hydrogen bonds, it can fold in a certain way called a beta pleated sheet, which is like that, which is made from the beta form. Or it could form like an alpha helix here, which is going to be a branched kind of structure where the hydrogen bonds are coming off like that, where my mouse is here. And that's going to be called an alpha helix here. So these two things, beta pleated sheet and alpha helix, are part of the secondary structure here. And the hydrogen NH has a slight positive charge, and the oxygen in the CO has a slight negative charge, obviously due to electronegativity. And hence hydrogen bonds are formed and hold the secondary structure here. Alright, cool. Tertiary structure here. So the tertiary structure is just the further folding of the secondary structure here. So to form a 3D shape, yeah. And this now has disulfide bridges, yeah, ionic bonds, and hydrogen bonds. You need to memorize that the tertiary structure has these three things. It has hydrogen bonds. For the secondary one was just hydrogen bonds. This one has ionic bonds, hydrogen bonds, and disulfide bridges, yeah. And the tertiary structure is always going to be unique. That's why when you talk about proteins, you say that when you talk about enzymes, yeah, you talk about their tertiary structure, or you talk about the tertiary structure has changed, that kind of thing, because that's the unique part, like the active site, yeah. So obviously here we've got tertiary structure. So that's just the further folding of the secondary structure to form a 3D shape. You need to mention, when you talk about the definition, yeah, you need to mention that it forms a 3D shape, and you need to mention the bonds that it's made of here, yeah. Alright, calm. So what it is just the alpha helix and the beta pleated sheet. That's all it is. Further folded, just made together. And a protein made up of more than one polypeptide chain. That's going to be quaternary. So quaternary is when you start getting more than one. So we made an alpha chain, we made a beta chain, they're tertiary, and we mix them together. Yeah. So as you see, hemoglobin has two alphas and two betas, and you might also have a prosthetic group here, which is like a group that's not protein. It could be like an iron group here, for example. We've got Fe2 plus. That's called the heme group. So that's called a prosthetic group here, because obviously prosthetic means something that's not from the body, and it means something external. Yeah. And obviously, because it's not a protein, yeah, it's a iron group here. That's why they call it the prosthetic group here. So that's what you also might have in the quaternary structure, yeah. Remember though, quaternary is only when you start mixing shit, yeah? When it's just the same shit, it's still tertiary, yeah? Alright, calm. Globular proteins and fibrous proteins, yeah? So this is a very small part of the course here. It's not really well covered like that, but you do need to know about that. Globular proteins are spherical and compact, so and they're involved in metabolic processes. So these are things like enzymes or hemoglobin, yeah? Those are going to be globular proteins, yeah? Whereas fibrous proteins, yeah, they form long chains or fibers. They're insoluble in water and they're useful for support, such as the collagen in the skin, yeah? So just make sure you know that. Cool, enzymes, bro. Enzymes, yeah. What you know about enzymes, bro? So enzymes increase the rate of reaction by lowering the activation energy, yeah. That's bait. That's how they work, yeah. So you got a bit high activation energy, yeah. You want to lower that, so you're gonna use an enzyme to lower that activation energy, yeah, to find an alternative reaction pathway, yeah. So they are a 3D tertiary globular protein whose shape is determined by the primary order of amino acids. So they're determined that by that primary structure, yeah. And enzymes are specific and can only bind to substrates that are complementary in shape. Complementary means it fits it nicely. So if they have a similar kind of shape, it will fit inside the groove nicely, yeah? So here we're going to talk about the induced fit model, yeah? So in GCSE, they talk about it's like an exact fit, yeah? Here we got the induced fit. So how the induced fit works, I'm just going to talk for a bit and tell you, man, how it works. So what happens is you got an enzyme, yeah? And of course, in life, nothing is perfect, yeah? So, of course, the substrate is not exactly going to fit the enzyme. As you see, it's a square thing. There's square, yeah? And that's going to be a triangle. So, because that's a square, yeah, it's not going to fit the enzyme perfectly, is it? So, therefore, what's going to happen is it's going to push into the enzyme, yeah? Pause. It's going to push into the enzyme and it's going to widen the thing. Pause again, yeah? It's going to widen it and force its shape into there. And because it forces the shape into there, the enzyme is gonna force itself back onto that shape. So it's gonna create stress. Do you see that? It's gonna create stress on that shape because it forced itself inside. So it's gonna be very tight. So the enzyme is gonna cause stress on the bonds in that square. So because of the stress that's caused onto that, it's gonna break and it's gonna release. Do you see that? So we call these enzyme substrate complexes, yeah, or ES complex, yeah. So here's another example here. So we've got the substrate. The active site is not perfectly complementary, but it's still complementary. So it's still complementary to it, but it's not perfectly exact same shape. And as you see, when it comes inside, it's going to force it to have the same kind of shape. It's going to force inside, and that's going to put strains on the bonds in the substrate. So therefore, the substrate's activation energy, the substrate's energy is going to reduce, so it's easier to break. So do you see how enzymes work here? So the active site is induced. It slightly changes shape to accommodate the substrate, which in turn puts strain on the bonds in the substrate and lowers the activation energy. So that's how enzymes work, yeah? Obviously, bro, this is bare long, bro, but just, write it down, make note of it, bro. Obviously, as temperature increases, the rate of reaction increases because higher temperatures provide more kinetic energy. Of course, this kinetic energy increases the frequency and force of collisions between enzymes and substrates, leading to more successful reactions, yeah? Always when you talk about this kind of shit, you need to say more successful reactions, yeah? More successful collisions, more successful reactions, yeah? So because there's a high kinetic energy here, what it means is that there's going to be more collisions between the enzymes and the substrates, which means there's going to be more successful reactions in it. So enzymes have an optimal temperature at which they function most efficiently. Yeah. So depending on where the enzyme is located, it's going to have a different optimal temperature. This temperature varies depending on the enzyme's origin and the organism it comes from. For most enzymes, in the human body, the optimal temperature is around 37 degrees. That's some good general knowledge you can know. However, beyond the optimal temperature, enzyme activity begins to decline rapidly. This is because excessive heat, yeah, disrupts the weak bonds such as hydrogen bonds and van der Waals forces. So obviously we've got intermolecular forces within the enzyme, like hydrogen bonds and van der Waals forces. And obviously at higher temperatures or temperatures that are above the optimal temperature, that's going to cause them to denature. It's going to cause them to start fucking around and start breaking apart. You get me? And that's not going to maintain that three-dimensional structure, because as we talked about, the three-dimensional structure, the tertiary structure, required those hydrogen bonds. So because they're broken now, it's going to collapse the whole situation. So denaturization occurs when the enzyme loses its native structure, rendering it unable to bind to substrates effectively. Denatured enzymes lose their catalytic activity permanently. So because the active site is going to change shape now, it's going to start falling apart. It can't react anymore, bro. That active site can't be used. It can't clock onto nothing. You get me? So it's going to be denatured. So this is a kind of curve. As you see, optimum temperature, as you gain kinetic energy, is going to increase. Optimum temperature is there. Anything past that starts denaturing. Yeah? pH. Extreme pH values can denature proteins, yeah, including enzymes, by disrupting the electrostatic interactions and hydrogen bonds that stabilize their tertiary structure. So that tertiary structure is held together by electrostatic interactions like CO, OH, delta positive, delta negative electronegativity. Yeah, but obviously the hydrogen and the hydrogen bonds, but obviously that's going to break down, which is going to cause a loss of enzyme activity, yeah, as the site becomes distorted or inaccessible. Enzymes have an optimal pH at which they function most effectively. This pH obviously depends on where the enzyme is. If it's in an acidic place, it's going to be like around 2. If it's in like the stomach, if it's not in the stomach, like the small intestine, it's going to be around 7 to 8, yeah. So it depends on where it's located, obviously. And it will have this kind of thing. So let's say the optimum is at 8. Anything too low than that, too acidic, it's going to denature because the hydrogens and shit are going to interfere with it. And anything too high is also going to denature. Enzyme concentration, yeah? So generally, as the enzyme concentration increases, the rate of reaction also increases. This is because you got more enzymes, so the enzymes can therefore react with the substrates that didn't have anyone to pair with, and they're gonna break it down. So more enzyme molecule available to bind with substrate molecules and catalyze the reaction. However, there comes a point where further increases in enzyme concentration do not increase the rate of reaction, as now there are more active sites, there's more enzyme now than substrates. So the substrates become the limiting factor. You need more substrates now to balance the active sites. You get what I mean? So this creates like a plateau. So as you're starting here, there's bare substrate and there's little enzyme, yeah? So as you're adding more enzyme, that substrate that's in excess is going to react with it. But as you keep adding enzyme and enzyme and enzyme, eventually it's not the enzyme that's in the limiting factor anymore. Now it's in excess. We've got too many enzymes now. We need more substrate. There's not enough substrate, so it's going to finish the whole reaction. It's going to reach a plateau, yeah? Alright, calm. Substrate concentration. So this is the same shit. As the concentration of substrate increases, the rate of reaction it will increase because again, there's an excess of active sites, excess of enzymes. So we're increasing the substrate, so substrate is going to react with those enzymes. However, at a certain point, the enzyme concentration becomes a limiting factor. So at a certain point, yeah, there's too much substrate now. There's not enough enzyme, so the rate of reaction no longer increases. Yeah. So you can just see that same kind of plateau there. Yeah. Inhibitors, yeah, called competitive inhibitors and non-competitive inhibitors, yeah. So let's talk about competitive ones, yeah. So competitive inhibitors, yeah. Inhibitors have the same shape as the substrate. So because they have the same shape as the substrate, it's going to be complementary to the enzyme, yeah. And it will bind to the active site and hence prevents the formations of enzyme substrate complexes. You need to use this kind of wording in the exam. Yeah? You need to say that it prevents the formation of enzyme substrate complexes. Like, trust me, that's what they're trying to see. However, this is reversible. So if you have a competitive inhibitor, yeah, it's actually reversible. If you add enough substrate to it, it will flood out the inhibitor, yeah. So here we got an inhibitor, yeah. It's the same shape as the active site there. So it's gonna come in there and it's gonna block the substrate from joining. Do you see how the substrate can't join anymore because my man took the situation. He entered it. So that's why it's gonna compete for the active site with the substrate. But if you put enough substrate, it can knock it out and it can come inside. You get me? Whoa, okay, cool. Non-competitive, yeah. So non-competitive inhibitors, yeah, they bind to an enzyme at a place called the allosteric site, yeah. Allosteric is when it's not the active site, it's another site, like another little crack or something on the enzyme. And so this inhibitor will come into there, it will force its way in, and it's gonna fuck up the whole situation. Look, that's gonna cause it to straighten out. that inside gap is going to be pulled inside, and now nothing can bind to the active site. So this is an issue, yeah. It cannot, it's not permanently, it causes permanent deformation, yeah. So it can't even reform. If you put bare substrate, it's not gonna do shit. You get me? It's peak, fam. So obviously, non-competitive inhibitors bind to the enzyme at the allosteric site, which is just a location other than the active site. This binding causes a conformational change in the enzyme structure, altering its active site's shape. Even if more substrate is added, the active site is now deformed and it cannot reform, yeah. Alright, cool. DNA. Okay. DNA. DNA stands for deoxyribonucleic acid, yeah? It's a molecule found in cells of living organisms, yeah, including humans, yeah? Think of DNA as the blueprint, yeah, like Jay-Z, or instruction manual for building the operating limit living organisms, yeah? It contains all the information necessary for an organism's growth, development, and functioning, yeah? DNA has a double helix structure which looks like a twisted ladder or a spiral staircase. So listen, fam, DNA is made of some bases. The bases DNA is made of is ATCG, adenine, thymine, cytosine, and guanine, yeah? And it's held together by a phosphate backbone. So we're going to talk about that. Let's talk about these bases or nucleotides, yeah? So a nucleotide is made of three things. It's made of a phosphate group, it's made of a pentyl sugar, and it's made of a nitrogenous base. So you need to know these three things. And so it's a polymer of DNA is a polymer of nucleotides. So this is one nucleotide. When you have bare together, that's going to be a polymer of nucleotides, yeah? So nucleotides consist of a phosphate group for the backbone, a deoxyribose sugar, that's the pentyl sugar, deoxyribose. That's why DNA is called deoxyribose nucleic acid, because you got this acid base, and you got the obviously, yeah. And there are four bases, yeah. Adenine, thymine, cytosine, and guanine, yeah. The bases pair up in a specific way. So A always pairs with T, forming two bonds. So AT and CG. Remember that. AT forms two bonds, CG forms three bonds. This is called complementary base pairing, yeah? So the complementary bases are paired by hydrogen bonds, yeah? And the phosphate backbone is held by phosphodiester bonds. So this phosphate group, this nucleotide will join to another nucleotide below it. And that's going to be formed together by a phosphodiester bond. That's the name of the bond between those phosphate groups, yeah? And on the other side, you'll have another nucleotide that way, which will be like, I'll show you here. You have another nucleotide there, which will be bonded to that. So this is adenine, which is bonded to thymine, and they will have two hydrogen bonds between them because AT, whereas CG will have three between them, yeah? So you just need to know about purines and pyrimidine, yeah? There's nothing really that you need to know here, but you just need to know that A and G are two-ring purines, that's what they're called. And T, C, and U, so that's uracil for U, that's the ribonucleic acid, are one-ring pyrimidines, yeah? So you just need to know what they are, you don't need to know anything about them. So RNA. So RNA is like DNA, but RNA stands for ribonucleic acid, yeah. So like DNA, it's a molecule found in cells of living organisms, yeah, and it plays a crucial role in protein synthesis and transfers genetic material, yeah. So like DNA, RNA is made up of nucleotides. Each nucleotide consists of three components. We've got a ribose sugar, a phosphate group here, and a nitrogenous base. So the difference here is we've got a ribose sugar instead of a deoxyribose sugar. So deoxyribose is the sugar in DNA, ribose is the sugar in RNA. That's why it's DNA and RNA. The nitrogenous bases in RNA are a bit different. They're adenine, A, C, G, and instead of T, we've got U now. So A bonds with U, C bonds with G. So it's the same shit as in DNA. It's just instead of T, we call it U, uracil, yeah? So it just replaces thymine. Also, it's one-stranded as well. It's not two-stranded. Okay, cool. Semi-conservative replication, yeah? If I just go back there, yeah? As you can see, it's obviously one-stranded. You got those situations, and that's all that you need to say there. Semi-conservative replication. So how does DNA actually get formed, yeah? Okay, so we got one. So the reason it's called semi-conservative, I'll talk about that in a minute, and it's just replication of DNA. So I've got one DNA strand. I want to make two DNA strands. How do I do that, yeah? So we've got the DNA here. You're going to use DNA helicase here. DNA helicase is going to break the hydrogen bonds that are between these bases here. So DNA helicase is going to come through and it's going to unzip, it's going to unwind the hydrogen bonds that are between the bases here. And this is going to separate the two strands. So this is going to basically split in half like that, yeah? And this is done by DNA helicase, which is an enzyme, yeah? Helicase, you can think of it as like hydrolysis kind of thing. Helicase, like breaking, yeah? That kind of shit. Cool. So one of these strands will get used as a template. So one of these strands, both of them actually, yeah, like but one on each side, is going to get used as a template. And now in the nucleus here, we've got bare free nucleotides. So you see these nucleotides, they're just chilling. They're doing nothing. They're just chilling inside. And so what's going to happen is when it unzips and you got these exposed nucleotides here, let's say this A1, a T will come from there and it will bond to the A. As you see there, T has bonded with the A. This G is going to bond with that C. Another G is going to bond with that C. Another one's going to bond with that. And it's going to come together. Do you see that? They're going to start bonding from hydrogen bonds. So one of these strands acts as a template. And the free nucleotides, these are not nucleotides that are falling out, by the way. These are two strands that have been separated. And nucleotides are coming in now and bonding to these to the nucleotides on each side. Do you see that? And now, once the nucleotides are bonded like that, as you see, uh, once the pairs are joined, DNA polymerase, so this is DNA polymerase, so it's polymer, yeah, so it's creating the polymer, is going to come and just wind that back up. So it's going to create bonds between these phosphate groups, the purple thing, yeah, and form phosphodiester bonds between them and just zip that up and connect it and make it one fluid motion. Do you see that? So make it one fluid motion there. So they've already bonded, the groups have already bonded, yeah, using hydrogen bonding, and that's just going to zip them up from the right hand side. Do you see that? And so now you're left with two identical DNA strands. Yes, identical because obviously one half got taken, and because it's complementary, it's going to make the same shit. So as you see here, we start off with one DNA strand. That one DNA strand got unzipped. Half of that DNA strand bonded with some free nucleotides, that were just walking around. The other half also bonded with free nucleotides. DNA polymerase came to zip both of them back up, and now we're left with two DNA strands here. So two identical DNA strands are formed. This is semi-conservative, as half of each strand contains genetic information from the original strand. Obviously, half of this strand was the original DNA, half of this strand was the original DNA, because remember, we used one DNA strand here and we split that in half and then we created these two. You see, so that's why it's called semi-conservative, yeah, conservative because it stayed the same. Come, let's talk about Molson's style, yeah. So bacteria were basically grown. This is basically an experiment that they did, yeah, to prove that DNA was semi-conservative and that it remained half of the shit, yeah. Molson style experiment, yeah. So the bacteria were grown in a medium with heavy hydrogen 15 for many generations, yeah. So nitrogen 15 is heavy, it's more than usual, yeah. And then some of these bacteria that had the heavy nitrogen 15, they got moved into an area with bare nitrogen 14, which is way lighter, yeah. Samples were extracted after 1 or 2 cycles of DNA replication. So these bacteria underwent DNA replication, and some samples were extracted, yeah. They did the centrifugation, which we'll see in topic 2. It's basically when you put it in this machine that spins it and it extracts it. And they found that the DNA weight was between heavy and light. Hence, each DNA has one heavy strand from the original and one light strand from the new. This supports semi-conservative replication. So if you didn't understand that, I'm going to say it in a normal way, yeah. Basically, yeah, there was some bacteria got put in a nitrogen 15 environment, which is a heavy nitrogen. So they got put in heavy nitrogen, and so that nitrogen was obviously used to make DNA. So they did DNA replication with the heavy nitrogen, and then some of that bacteria was moved to the light nitrogen. If yeah, if it was not semi-conservative, then it would have just been light because they got moved to the new light area, so it would just been the new nitrogen that's used. But because it's semi-conservative, some of the older nitrogen, the heavy nitrogen from the first environment, was also used because that first DNA strand was heavy, and then the new DNA strand is going to be light because of the semi-conservative replication. So that's why when they found the weight was between heavy and light, which means that both nitrogen 14 and nitrogen 15 were both used, not just nitrogen 14. So that proves, yeah, that DNA replication uses half of the original strand and it doesn't just use new strands. You understand? So it proves that DNA is not just created, it proves that it's like recycled. It proves that the DNA strand is split by helicase and adds another strand to that. You get me? So that's what that's saying. ATP, yeah. So ATP stands for adenosine triphosphate. It's a nucleotide derivative. As you see, adenine is the same as like adenosine, yeah. So it comes from adenine, yeah. And it consists of ribose, adenine, and 3 phosphate groups. So it's similar to a nucleotide, really and truly. It's got ribose, which is nucleotide and RNA also has. It's got adenine, which a nucleotide and RNA also has. But instead of 1 phosphate group, it's got 3 phosphate groups, yeah. So energy is released when ATP is hydrolyzed, yeah. So ATP, they call it the currency of biology, yeah. They call it the money of biology. It's used, it's exchanged, that kind of thing. It's like the main energy source here. So energy is released when ATP is hydrolyzed to form ADP and the phosphate molecule. So ADP is adenine diphosphate, yeah. So that's adenine with 2 phosphates. So ATP is broken up, yeah. is hydrolyzed to form ADP and one phosphate molecule. This is catalyzed by ATP hydrolase. So that's the enzyme that breaks that up. It does the hydrolysis. That's why it's called ATP hydrolase. Yeah. The inorganic phosphate can be used to phosphorylate other compounds and make them more reactive. So, as well, another use of ATP is to make other things more reactive, and it does that by basically adding the inorganic phosphate group to other molecules, which makes them more reactive. Yeah, that's called phosphorylation. Yeah, and condensation of ADP and inorganic phosphate. So when you mix ADP and phosphate together, that's catalyzed by ATP synthase because you're making ATP here, so synthesis. That's going to produce ATP, yeah, during photosynthesis and respiration. So just a little recap there. All that's going on there is all you need to know here is that ATP hydrolase breaks ATP up into ADP and P, and that's going to create energy. That's going to produce energy, and ATP synthase does a condensation reaction which mixes together ADP and P to make ATP. Yeah, and that obviously uses energy. Okay, cool. Properties, yeah. So ATP is bro, I said that in a weird way. Alright, cool. So ATP is an immediate source of energy, yeah. So you need to know that that's an advantage of ATP. It's an immediate source of energy and is more desirable to use than glucose, yeah, as ATP can be broken down in a single step. All you need is that ATP hydrolase and it just breaks ATP like that. ATP gets snapped into ADP and P, yeah, and you release energy. And it also releases manageable quantities of energy, yeah. So not too much, not too little, manageable, yeah. So that's what you need to write down. This is important. They'll ask you, explain the advantages of ATP. ATP isn't stored in large quantities as well, so it can be easily reformed from ADP in seconds, yeah. And ATP is used in a variety of different ways, so it's very versatile, yeah. It can be used in metabolic processes, it can be used in movement, active transport, and secretion, yeah. Alright, cool. Water, yeah. So water is a metabolite. So you need to know the 5 properties of water, yeah? So there's 5 properties of water that you need to know. First of all, it's a metabolite, yeah? All that means is that it's used in condensation and hydrolysis. In hydrolysis, you're using water to break up a molecule, and in condensation, you're using water you're releasing water when you make a molecule, yeah? So that's all it means by metabolite. It just means it's like an intermediate, yeah? It's also a solvent, yeah? Which allows gases to readily diffuse as well as enzymes and waste products, yeah? So it's a solvent as well. That's the second situation. Yeah. Also, it has a high specific heat capacity, which helps reduce temperature fluctuations and acts as a buffer. Specific heat capacity, yeah, means how much energy a substance can take before it increases by 1 degree. So what this means here is that water, as you see, like in the ocean, if you ever been to an ocean, yeah, Brighton thing, yeah, as you see outside in the sea, the water is always bare cold in it. So why do you think that is? Even if it's a bare sunny, even if there's sun, there's whatever, yeah. You get me? Rice lip Lido thing, yeah. Why is it always that the um water is cold? It's obviously because it has a high specific heat capacity, so it doesn't matter. You could put all that heat onto it, it's not going to increase the temperature. You can add a lot of energy to water, and the temperature is not going to increase. You get me? That's what it's trying to say. So you see the situation. So that's what I mean. So it acts as a buffer. It's a good temperature buffer. It keeps the temperature the same. It doesn't increase in temperature that easily, yeah. So also it has a higher latent heat of vaporization. So what that means, yeah, is it takes a lot of energy. It's like specific heat capacity, yeah. It takes a lot of energy for it to vaporize. So it requires a lot of energy, yeah, for it to move from a liquid into a gas. And what that means by provides a cooling effect is it means that once you do start sweating, yeah, once the sweat vaporizes and it turns into a gas, yeah, it's gonna carry away a lot of energy with it because it took bare energy, yeah, to move it from liquid to gas. So that means that all that energy that got put into it to turn it into a gas, it's gonna release all that energy with it when it turns into a gas. So that provides a cooling effect here. And obviously cohesion and adhesion as well, they allow for transport of water in the xylem, yeah. So cohesion is basically when water molecules, because they have hydrogen bonds, they can kind of stick together. That's cohesion, yeah. They stick together and that allows them to move quicker, move in a faster way. That's like, for example, when you have the an adhesion as well. That's like, for example, when you got like you're sitting in a lab or whatever, and you got like water dripping from the tap or wherever you are, yeah, you got water dripping from the tap. As you see, it takes a long time, yeah, for it to drip down. That's obviously because the hydrogen bonds between the water molecules, yeah, they're causing it to stick together. That's called adhesion and cohesion. Cohesion is when they stick together. Adhesion is when they stick onto surfaces, like they're sticking onto the metal of the tap, yeah. You get me? So, and that allows for transport in the xylem, which you'll see in the future as well. So yeah. Inorganic ions, yeah. So hydrogen ions determine the pH. The higher the concentration of hydrogen ions, obviously the lower the pH, the more acidic it is, yeah. The iron ions are used in hemoglobin, which carries oxygen in red blood cells. Sodium ions are involved in co-transport of glucose and amino acids, which we'll see in a minute, co-transport. And then phosphate ions are obviously a component of RNA and ATP, yeah. You got phosphate in DNA, you got phosphate in ATP, yeah. Okay, cool. The tests, I think this is the final thing, yeah. Alright, cool. The test here. So all monosaccharides and maltose are reducing sugars. So memorize that. Monosaccharides and maltose, they get reduced. They're reducing, yeah. So to do the test here, what you do is you add equal samples of Benedict's and the food sample, and they must be liquid. You need to remember this, yeah. So they must be liquid. Step 2, you heat the mixture in a water bath for 5 minutes at 100 degrees Celsius. And step 3, if it's positive, yeah. So if there is actually sugar that's found, if there is a monosaccharide found, a reducing sugar, it will go from blue to orange or brick red. Yeah. So the solution will go from blue after the water bath in 5 minutes for at 100 degrees. It will turn brick red. Yeah. So that's positive. Yeah. That means that there was sugar found. Memorize this. Write this down now. Memorize this now. You know what I'm saying. It will be good use later. Non-reducing. So other disaccharides or polysaccharides are not going to be reduced because they're made of bare shit. So other disaccharides apart from maltose, yeah, all the other disaccharides and like lactose and shit and sucrose and polysaccharides, they're non-reducing. So for this, yeah, if you do the first test first and a negative test will appear. Because the negative test appeared, it means it's non-reducing. It didn't get reduced. So you're gonna have to add another 2 centimeter cube of food sample and 2 centimeter cube of dilute hydrochloric acid, yeah? And then you place them in a hot water bath for 5 minutes. This is going to hydrolyze. So the dilute HCl will hydrolyze the disaccharides and polysaccharides into their constituent monosaccharides. So let's say we've got lactose, yeah? The HCl is going to break up lactose into glucose and galactose. And then those are now reducing sugars. So those will do the same test and they'll be calm. So then sodium hydrocarbonate is added to neutralize the test tube, as Benedict's won't work under acidic conditions. Because you added HCl, the Benedict's is not going to work. It's going to fuck up in it. So you need to add sodium hydrocarbonate here to neutralize that. And then the solution is retested with Benedict. So you do the same test as the first and you'll get a brick red color. So it will work now. So what we need to do is break up that polysaccharide here into the monosaccharides, then repeat the test. And to break them up, you need to use dilute HCl to hydrolyze it. And then you need to obviously neutralize that with sodium hydrocarbonate, yeah? Alright, calm. Emotion test, so this is for lipids, yeah? So again, you take a grease-free test tube. That's important. Always mention that in the test. Take a grease-free test tube because it might affect the results here. And add 2 centimeter cubed of sample and 5 centimeter cubed of ethanol. You then shake the test tube. It's important you say you shake it. You have to say that you shake it, yeah? You shake it to dissolve all the lipid. And then you always have to say add 5 centimeter cube of water and shake gently. Always add 5 centimeter cube of water after you do that, yeah? And what you'll have is you'll have a cloudy white color, which shows that there's lipid present. Obviously, it will just be see-through if there's nothing, yeah? Because you're just mixing ethanol and water. Alright, cool. So that will cause a white, cloudy white color, yeah? A white emulsion, yeah? If there's lipids, yeah? Alright, Biret, yeah? So this is used to test for presence of peptide bonds, yeah, so proteins, yeah, use Biret test, yeah. So you place a sample in a test tube with an equal amount of sodium hydroxide. So you place a sample in equal amounts with sodium hydroxide. Then you add a few drops of dilute copper sulfate solution and mix gently. This copper sulfate and sodium hydroxide is what makes the Biret reagent. So in A level, you normally need to explain the actual two things that make Biret. You can't just say I add Biret. You need to say that you mix sodium hydroxide and dilute copper sulfate solution, yeah. And it goes from blue, because obviously copper sulfate is blue, to purple. Then there's a peptide bond. So if there's a protein, then it will go from blue to purple, yeah? So all you need to do here is just add the sample with sodium hydroxide, and then add a few drops of dilute copper sulfate and mix. That's it. Starch test, yeah? So the food extract or solution is placed in a test tube or a spot plate, and a few drops of iodine solution is added, and it will obviously, which is orange. And if there's starch, it will obviously turn blue-black. Yeah, that's easy, bro. Yeah, that's just starch, yeah? Alright, and that's the end, man. That's the end of the whole chapter here. So obviously, go do the questions as well. Watch the question video, and you should be certain, man. That's literally everything covered, by the way, in this topic. Like even the things that are small, minute. If you memorize everything that's been said here, that's it, bro. You're next level.